Chapter 4. Playing God. Customized DNA Molecules. DNA molecules are immensely long. Only one continuous DNA double helix is present in any given chromosome. Popular commentators like to evoke the vastness of these molecules through comparisons to the number of entries in the New York City phone book or the length of the river Danube. Such comparisons don't help me. I have no sense of how many phone numbers there are in New York City, and mention of the Danube more readily suggests a Strauss waltz than any sense of linear distance. Except for the sex chromosomes X and Y, the human chromosomes are numbered according to size. Chromosome 1 is the largest, and chromosomes 21 and 22 are the smallest. In chromosome 1, there resides 8% of each cell's total DNA, about 250 million base pairs. Chromosomes 21 and 22 contain some 40 and 45 million base pairs, respectively. Even the smallest DNA molecules, those from small viruses, have no fewer than several thousand base pairs. The great size of DNA molecules posed a big problem in the early days of molecular biology. To come to grips with a particular gene, a particular stretch of DNA, we would have to devise some way of isolating it from all the rest of the DNA that sprawled around it in either direction. But it was not only a matter of isolating the gene. We also needed some way of amplifying it, obtaining a large enough sample of it to work with. In essence, we needed a molecular editing system, a pair of molecular scissors that could cut the DNA text into manageable sections, a kind of molecular glue pot that would allow us to manipulate those pieces, and finally, a molecular duplicating machine to amplify the pieces that we had cut out and isolated. We wanted to do the equivalent of what a word processor can now achieve, to cut, paste, and copy DNA. Developing the basic tools to perform these procedures seemed a tall order, even after we cracked the genetic code. A number of discoveries made in the late 60s and early 70s, however, serendipitously came together in 1973 to give us so-called recombinant DNA technology, the capacity to edit DNA. Well, this was no ordinary advance in lab techniques. Scientists were suddenly able to tailor DNA molecules, creating ones that had never before been seen in nature. We could play God with the molecular underpinning of all of life. This was an unsettling idea to many people. Jeremy Rifkin, an alarmist for whom every new genetic technology has about it the whiff of Dr. Frankenstein's monster, had it right when he remarked that recombinant DNA rivaled the importance of the discovery of fire itself. Arthur Kornberg was the first to make life in a test tube. In the 1950s, as we have seen, he discovered DNA polymerase, the enzyme that replicates DNA through the formation of a complementary copy from an unzipped parent strand. Later, he would work with a form of viral DNA. He was ultimately able to induce the replication of all of the virus's 5,300 base pairs of DNA. But the product was not alive. Though identical in DNA sequence to its parent, it was biologically inert. Something was missing. The missing ingredient would remain a mystery until 1967, when Martin Gellert at the National Institutes of Health and Bob Lehman at Stanford simultaneously identified it. This enzyme was named ligase. Ligase made it possible to glue the ends of DNA molecules together. Kornberg could replicate the viral DNA using DNA polymerase, and by adding ligase, joined the two ends together so that the entire molecule formed a continuous loop. just as it did in the original virus. Now, the artificial viral DNA behaved exactly as the natural one did. The virus normally multiplies in E. coli, and Kornberg's test tube DNA molecule did just that. Using just a couple of enzymes, some basic chemical ingredients, and viral DNA from which to make the copy, Kornberg had made a biologically active molecule. The media reported that he had created life in a test tube, inspiring President Lyndon Johnson to hail the breakthrough as an awesome achievement. The contributions of Werner Arbour in the 1960s to the development of recombinant DNA technology were less expected. Arbour, a Swiss biochemist, was interested not in grand questions about the molecular basis of life, but in a puzzling aspect of the natural history of viruses. He studied the process whereby some viral DNAs are broken down after insertion into bacterial host cells. Some, but not all, otherwise viruses could not reproduce, host cells recognized certain viral DNAs as foreign and selectively attacked them. But how and why? All DNA throughout the natural world is the same basic molecule, whether found in bacteria, viruses, plants, or animals. What kept the bacteria from attacking their own DNA, even as they went after the viruses? The first answer came from Arbour's discovery of a new group of DNA-degrading enzymes, restriction enzymes. Their presence in bacterial cells restricts viral growth by cutting foreign DNA. This DNA cutting is a sequence-specific reaction. A given enzyme will cut DNA only when it recognizes a particular sequence. EcoR1, one of the first restriction enzymes to be discovered, recognizes and cuts the specific sequence of bases, GAATTC. But why is it that bacteria do not end up cutting up their own DNA in every place where the sequence GAATTC appears? Here Arbour made a second big discovery. While making the restriction enzyme that targets specific sequences, the bacterium also produces a second enzyme that chemically modifies those very same sequences in its own DNA wherever they may occur. Modified GAATTC sequences present in the bacterial DNA will pass unrecognized by EcoR1, even as the enzyme goes its marauding way, snipping the sequence wherever it occurs in the viral DNA. The next ingredient of the recombinant DNA revolution emerged from studies of antibiotic resistance in bacteria.